Hello friends, in this video lecture I will talk about the structure of pollen grain. The pollen grains are produced inside the anther sac, and a pollen grain is the first cell of male gametophyte because it ultimately produces the male gametes. We know a pollen grain produces two male gametes that helps in plant reproduction. So let me to write the point. So it is the first cell of male gametophyte. The pollen grain is the first cell of male gametophyte as it produces the male gamete. And the branch in which we study the pollen structure morphology is known as palynology. So palynology is the branch in which we study the structure of pollen grain. So I have drawn the structure here. So look at the structure. A pollen grain is globular in outline. So it is globular or spherical. It is globular or spherical in, that is globular means spherical in outline, globular in outline. Other shapes of pollen grains are also found, and its diameter is 25 to 50 micrometer. And this pollen grain has a highly resistant wall. It has a covering, the wall covering, it is highly resistant. And the cytoplasm of the pollen grain is rich in starch. So the pollen grain cytoplasm is rich in starch and unsaturated oils. So the function of this unsaturated oil that it helps the chromosome of the pollen grain from radiation damage. So the nucleus that contains the chromosome, the unsaturated oil helps the chromosome of the pollen grain from radiation damage. Means it helps from UV radiation. And the resistant wall, this wall is called sporoderm. So the pollen grain wall is known as sporoderm. And this wall of sporoderm is made up of two layers. This sporoderm wall is made up of two layers, that is outer exine and inner intine. So now we will see the composition of intine. So the intine is made up of cellulose and pectin. So it is pectocellulosic nature. So it is pectocellulosic in nature. Then so come to exine. So this exine is the outer layer of the pollen grain, is the outer layer of the sporoderm, that is exine. So exine is made up of a highly resistant fatty substance. So the exine is made up of a highly resistant fatty substance. So this is important. So the substance is sporopollenin. And this sporopollenin is not degraded by any enzymatic actions, or it is not affected by high temperature, strong acids or alkalis. So this sporopollenin makes this wall resistant. So due to this resistant nature, the pollen grains are well preserved as microfossils. So we can store pollen grains for long period in the form of microfossils. Further, the exine is differentiated. So now we'll study the structure of exine. The exine is further differentiated into three layers. This is the exine, and this exine layer is differentiated into an inner, a continuous foot layer. This foot layer is continuous layer, and a middle discontinuous, that is baculum, which is a discontinuous layer, and this foot layer is continuous layer, and the topmost, that is the tectum layer. This is also discontinuous, this tectum layer, and this tecton layer, this is the outermost layer of the exine. So it provides a characteristic sculpturing. It provides a characteristics sculpturing and designs to the surface of the pollen grain. So this is about the structure of pollen, the structure of exine, and the intine is made up of cellulose and pectin, and exine is composed of sporopollenin. This is a highly resistant fatty substance. The exine is further differentiated into three layers, and the topmost layer, that is the tectum. So in insect pollinated pollen grain, the exine is covered over. The exine is covered by a yellowish viscous sticky substance, yellowish oily sticky substance known as pollen kit. So in entomophilous pollen grain, or the pollen grains those are carried out by insects, the exine is covered by a yellowish viscous, that is oily sticky substance known as pollen kit. Pollen kit. So these pollen kits are made up of, the pollen kits are composed of lipids and carotenoids. Lipids and carotenoids. So what is the function of pollen kit? So due to its sticky nature, means the pollen kit is sticky. So the pollen grains easily adheres to the mouth or the body surface of the insects. That that facilitates the transport of pollen grain from flower to flower, and it also helps from UV radiation. So this pollen kit, since it is composed of lipids and carotenoids, so it also protects from UV radiation. UV radiation. Certain places, so look at this diagram, at certain places the exine is absent. So at three places the exine is not continuous, the exine is absent. So in this site, the exine, in this place, either the intine is thickened, there is thicker intine, or in that place there is deposition of callose. So in these regions, either the intine is thicker, or there is deposition of callose, and this site are known as germ pores. So this region is called a germ pores. So what is the function of these germ pores? So we know a pollen grain falls on the stigmatic surface, falls on the stigma. Then the pollen grain, if it is a compatible pollen grain, then the pollen grain starts to germinate. Then the intine comes out, the protrudes out in the form of a pollen tube through the germ pores. So the function of germ pores that it helps in. So this is the region, this is the site through which the intine comes out. The intine comes out in the form of a pollen tube. So the germ pores helps in the process of pollen germination at the time of sexual reproduction. And the number of these germ pores, so the pollen grain of the dicot, and the tricolpate means, so there are three germ pores found in dicot. And in case of monocot, the pollen grain is monocolpate. There is the presence of only a single germ pores that is found in the pollen grains of monocotyledons. So the Greek kolpos means germinal furrows or apertures. So in dicot plant, the pollen grains are tricolpate, and in monocot, the pollen grains are monocolpate. Means only a single germ pore is found in the pollen grains of monocotyledon. So from the very beginning, the pollen grain is a uninucleate. It is a uninucleate haploid cells from the very beginning. But the structure here, the pollen structure here shows it is two cell structure. So it is a two cell structure. So the larger one is the vegetative cell. The larger one is the vegetative cell, and the smaller cell, that is the generative cell. Generative cell. So when the pollen grains become mature, or at the time of liberation, the pollen grain is found either in two cell stage or in three cell stage. So this is the larger. The vegetative cell is the larger of the two. It has a large irregular shaped nucleus, and its cytoplasm is rich in abundant reserved food substance. And the generative cell, it is smaller and it is spindle shaped, and the generative cell floats in the cytoplasm of the vegetative cell. So it is a two cell pollen grain. So at the time of liberation, the pollen is either two cell or three cell. So in certain plants, this generative cell nucleus divides to form two male gametes. So after division, when it produces the two male gametes, then each male gamete surrounds some cytoplasm around it and becomes two cells. So the pollen grain becomes three cell. So in 40% flowering plant, at the time of pollen liberation, the pollen grain is found in three cell stage. So this is about the structure of pollen grain. I hope this video will help you understand the structure of pollen grain. Thanks.